Hello and welcome to Organogenesis. Organogenesis is the process of initiation and development of plant organs such as root and shoot from an undifferentiated mass of cells. There are two main developmental pathways induced by plant growth regulators in plant tissue cultures. It is organogenesis and somatic embryogenesis. Organogenesis is the generation of organs from tissues, example leaves, callus. These regenerated organs may be shoots, roots, or leaves, each with a meristem at one end of the axis but not on the other end. Somatic embryogenesis is the generation of embryos from tissues. Embryos are bipolar structures with a shoot meristem at one end of the axis and a root meristem at the other end. Auxins and cytokinins are important plant growth regulators which induce organogenesis. The ratio of auxin to cytokinin control the formation of roots, callus, or shoots by organogenesis. An intermediate ratio of auxin to cytokinin induces callus. Low cytokinin and high auxin ratio induces the root formation, while high cytokinin and low auxin induces the shoot formation. In this prac exercise, you will study how different plant growth regulators induce development of different plant organs. Turn on the laminar flow and wipe it with 70% ethanol before use. Label your plates. Each group is assigned a treatment or control per plant from the following table. Group 1 will have only the basal media without any plant growth regulator. This will serve as a control. Group 2 will have basal media supplemented with 5.4 micromolar alpha naphthalene acetic acid. This is an auxin. Group 3 will have basal media supplemented with 4.4 micromolar of benzyl aminopurine. This is a cytokinin. Group 4 will have basal media supplemented with 44.4 micromolar of benzyl aminopurine and 0.54 micromolar of naphthalene acetic acid. This group will have both cytokinin and auxin as a treatment. Lastly, group 5 will have basal media supplemented with 10 micromolar of thiodiazuron, which is a cytokinin. If you remember from the prac exercise on somatic embryogenesis, we use thiodiazuron to induce somatic embryogenesis. Prepare the stock solution of plant growth regulators at a concentration of 5000 micromolars. You would need 95% ethanol to be used as a solvent to dissolve thiodiazuron and 1 molar NaOH to dissolve benzyl aminopurine and naphthalene acetic acid. Filter sterilize the plant growth regulator solutions. Add the plant growth regulators to the molten media. You can also add the plant growth regulators to the petri dishes. Pour the media into the Petri plates and swill gently. Allow the plates to solidify before use. Next, we will prepare the explants. We will use 4 to 6 healthy turgid leaves for organogenesis. You will be working in aseptic conditions. Turn on the laminar flow and prepare for exercise. The leaves are sterilized in 70% ethanol, followed by washing in 10% sodium hypochlorite for 15 minutes. Leaves are then rinsed with steriles 4 times, dispensing of used solution. Inside the laminar flow cabinet, cut the leaves into greater than 5 into 5 mm explants using a sterile scalpel and forceps. Be sure to remove the midvein from each leaf. Immediately place the explants abaxial side down, touching the media. The leaf on the right is showing the abaxial side. Once you have placed the abaxial side down on the prepared media, the treatments are to be left for incubation for 3 weeks time. This is the end of part 1 of the exercise. The organogenesis will be induced and new organs in form of shoots and roots will be developed. Welcome back to the part 2 of the exercise. Today you will record your observations. Based on the known facts of plant growth regulators, observe in each plate the organogenesis and development of root, shoot and callus. You will observe that high cytokinin induce the shoot formation, high auxin concentration induce the root formation, an intermediate ratio of auxin and cytokinin induce callus formation. Record your observations as shown in the table. Count the number of explants with callus. Count the number of explants with shoot. You will be provided with the class data to answer the questions and submit your assignment. Thanks for watching.